So my pleasure today to introduce Ivan Araujo from Pierce Laboratory, where he's a fellow and runs his lab. He's also an associate professor in psychiatry, our department, as well as molecular physiology here. And Ivan's work has really been across a whole range, and I think this comes out of his training and his background. And I mean a whole range, both technically, conceptually, and he's had an impact across many fields in his relatively young career. You're still young enough to say that. Right? It's getting close, man. I looked at you right after I said that. I said, yeah. So his graduate training was in Oxford with Ed Rolls, where he was doing human imaging studies, looking at really basic sensory inputs, looking at both taste, olfaction, all kinds of thirst, all kinds of imaging studies throughout multiple regions of the brain. And I remember seeing those papers and being really impressed that the amount of work really that was in them to look at all these different areas of the brain with these different modalities. And this had obvious implications for ingestive behavior. And he moved on to a postdoc with Miguel Nicolelis and Sid Simon at Duke, where his work became even more directly involved and impactful with ingestive behavior. And there he moved into animal or non-human animal models, so out of the imaging, and in that environment he became really an expert in recording and looking at encoding and deciphering encoding from recorded sets of neurons, again, throughout multiple regions in the brain. I was really, that paper that you had from there, I was amazed at how much analysis must have gone in. Parts brain I'm directly interested in, striatum, parts of the cortex. And he had several studies there, one looking at satiety that was really impactful in the field, looking at sort of the encoding of satiety as animals were getting to that state. So this is really basic and fundamental, impacting both how we think about behavior in general and specifically ingestive behavior. And this trend to do work that's pretty broad and impactful across fields has continued here in his studies, where he's really bridged between multiple fields. He's looked at sort of sensory input and how it affects ingestive behavior, but he's also looked at nutritive and post-ingestive feedback. And this is probably influenced, from his work, has influenced my views of food intake behavior the most, where we get these signals that come that are independent of sensory signals from the gut within minutes. Am I exaggerating? Within minutes? Milliseconds, not milliseconds, minutes. They come from the gut and they go right to dopamine circuits, for example, and influence them. So these are really, this gut-brain axis, which gets a lot of discussion. I think much of this field is now in need of mechanistic studies, and Ivan's work has done that, looking at signals that are coming right from the gut when nutrients come in, that signal directly to these reinforcement centers of the brain. And so this work, I think, has set the stage for the research going into gut-brain axis to get much more mechanistic and much more into specific neuroscience models within the brain. And as you can see from his title today, he continues to sort of branch out, which I think reflects his general interest in coding and how neural circuits influence behavior. And he's had a recent study, which he sort of looks like he's going to talk about today, looking at sort of the basis of aggression. Again, in a different part of the brain, completely different sort of context, but again, taking the same innovative approaches technically and conceptually and producing impactful work. So I want to welcome you and looking forward to the talk today.
So thank you, Rob, for this really nice introduction. I had forgotten about some of these studies. So I am getting old, yeah. So so I would just start by reminding you that, like, for some 400 million years ago, our ancestral vertebrate was essentially a fish that used it to eat pieces of food floating around the ocean. And this fish just hang around like a swimming, getting food into its mouth and into its gastrointestinal tract. But for some reason, a genetic mutation took place eventually into this animal, so that some of the more anterior gill arcs transformed into this articulated open around the mouth of the animal that we eventually called the jaw. So this small bone transformation in the head of this primitive fish had possibly the most dramatic implication for behavior and for the evolution of the nervous system that we can think of in terms of vertebrate evolution. So this small change in this gill arc of this animal produced a whole range of new opportunities for survival for this animal that eventually led for these fishes displaying this articulated aperture in the mouth to evolve into animals that moved into the land and so on until primates and us. So the reason why is this important, it's because this animal gained or expand its behavioral repertoire in a very dramatic way. So once jaws appeared in evolution, one new behavior that emerged was the ability to hunt or to kill other animals and use them as nutrition. So this ability to capture, kill, and digest other animals incredibly increased the opportunities for survival for those animals possessing a jaw, and also changed their whole behavioral setting, because these animals, of course, became more aggressive in terms of being able to, having the motivation to capture and kill other living beings. So, but in addition to being able to capture prey and digest them using the jaw, these animals had a third task to care about, which was to protect its territory from other animals possessing jaws that would be, of course, also willing to capture the same prey. So you can see this small modification of the vertebrate head completely changed the context in which animals survive and the kind of nervous system that they need to survive efficiently. So the complexity of the behaviors that emerged can be seen in simple movies like this, where you have a very fast, eager predator like this big feline pursuing its prey. So there is a very precise locomotion pattern triggered towards the prey. So at some point, the animals makes the decision to jump onto the prey and capture it. Then a whole range of very complex craniofacial behaviors are initiated with the twisting of the neck, the opening of the jaw, and the grabbing of the neck of the prey. So you can see that the capture and killing of prey present a range of very complex computational problems to this vertebrate brain that has to be solved in some way by some sort of very sophisticated neural circuit that evolved through evolution to control these new parts of the vertebrate head that just emerged. So fortunately, in the lab, we don't need these big felines, and because mammals evolved or appeared on Earth originally as insectivore, as insect hunters, this trait is preserved in a very wide range of mammals, including mice. So very interestingly, in laboratory mice, like the usual black 6 that we get in our labs, is even though it has been a laboratory mice for many, many, many generations, it still has its trait of seeing a cricket hanging around its cage, something they never seen before, and still they have this innate motivation to pursue it, kill it, and eat it. So the mouse is still an appropriate model for studying hunting behavior, even though we do not see it as a very fearful predator. So years ago, scanning studies of brain activity during hunting behavior in animals, in rodents, indicated that a region in the temporal lobe that we call the central amygdala is probably important for hunting behavior, along some other areas in the brain. So, so this is a region here located lower in the temporal lobe that is usually associated with emotional behaviors. You probably heard a lot about fear conditioning and the role of the central amygdala in this type of behaviors. But the central amygdala had a very interesting property in terms of neuroactivity. Its activity increased during hunting in a way that did not increase as much during food intake per se. So this region is, in principle, very interesting in hunting behavior because it seems to have some specificity to that behavior in particular. Another interesting aspect of the central amygdala that is somewhat forgotten these days is that in the old days, the central amygdala was considered to be part of the basal ganglia, that is the set of nuclei in the brain that control motor actions. And this is because developmentally, the central amygdala has a common origin along with the dorsal striatum. So if you go very caudal in the dorsal striatum, you can see that the amygdala, the central amygdala, is basically a continuation or a transition from a larger area that we call dorsal striatum. So there is a lot of motor connectivity associated with the central amygdala that is not as studied today as some other aspects of its functions, but probably has to do with the ability of animals to perform these complex behaviors that involve so many different motor systems. So to study the central amygdala and to activate these inhibitory neurons in the central amygdala in a somewhat specific way, we use a technique called optogenetics that is essentially linked to delivering a light-sensitive gene to neurons in the brain, usually using viruses. So we inject a virus into the brain of the animal that contains a gene that expresses, that encodes for a light-gated channel that when activated by blue light depolarizes the neuron. So in this way, because neurons don't normally respond to light, we can choose genetically which neurons to target, and we can use pulses of light to trigger activity in these neurons. So if you were to record these neurons and pulse light on them, you would see that their membrane potential changes very dramatically, very fast. It's a technique that works very well. So we started with like some screening of the effects of activating central amygdala in animals. So here is the optical fiber over the central amygdala that delivers light to the animal. This is a piece of wood stick. So we randomly turned on the light, and you can see that the behavior of the animals change very fast towards grabbing and capturing this piece of wood that they normally don't care about, and they start biting and sometimes hiding this object in a part of the cage. When we put another arbitrary object in the cage, you can see that the triggering of the laser produces this stereotyped behavior, which is moving towards the object, biting it, sometimes chewing pieces of it, and swallowing it as if it was a target that had some sort of intrinsic value. When we had animals, like bug-like toys moving around the cage, you can see that the animals are very annoyed by it, and so this experimental design was purchased on Amazon. So you can see the animal really doesn't like. But when we turn on the light, you can see that the animals, for some reason, decide to attack it as if it was something worth doing it. And for as long as the laser is on, they will bite and, let us say, try to kill this object as if it was a real insect that they would be interested in. You can see that the animals actively pursue the prey. So the prey is moving, Of course, this is the beginning. This is initially annoying to the animal, but once the light is on, you can see that the animals intentionally pursue the object. They go towards it with a very clear interest in it. And when we if we use other techniques that chronically activate this neuron in the central amygdala, that turn them on constantly for hours, we see just this behavior going on for a relatively long time. So you can see this animal here, observed from the top, pursues the object and bites it and is very eager into trying to immobilize the object. More generally, we observed a very strong disinhibitory effect on feeding behavior by activating the central amygdala. So we have animals in the cage around pieces of wood that, of course, they don't usually eat. When the light is on, you can see the light is being delivered to the brain. These animals would grab this piece of wood and start chewing it, and very often swallowing it. So not in all, but in a group of animals, we would find these pieces of wood stick present in the stomach. So they process the whole digestion of the object through the gastrointestinal tract. So in this case, they finish with the first one, and then they would go immediately for a second piece. And as soon as the laser is off, the animals will interrupt the behavior and go like what I was doing or something like that. So the behavior never takes place if the laser is off, of course. So the question is why? So why does the amygdala produce this type of very complex motor behaviors? So here is a picture of the brain. We injected some sort of tracer into the central amygdala region. And when we look into where it projects, this is a side view of the brain. The eyes would be here, and here's the back of the animal. So this is a side view. Here's the brain stem. And so these green areas are represent terminals arising from the central amygdala and terminating in these regions of the brain. So of particular interest are these terminals terminating here in the lower brain stem. I will zoom in a little bit this area. So this area is very interesting because this MO5 area contains the motor neurons of the trigeminal nerve, which is the nucleus that controls the movement of the jaw, the opening, the closing, and the strength of the jaw muscles. You can see that the amygdala doesn't project to the motor neurons themselves, but it projects all around this nucleus, this motor nucleus, and the seventh nucleus, which is the motor nucleus of the facial nerve. So it projects very heavily to this area called PCRT, which is the parvocellular part of the reticular formation. So the reticular formation is essentially all this area here, where premotor neurons, that is, neurons that are immediately connected to the motor neurons, are present. So for reasons that are going to become clear later, the amygdala gains or exerts a very powerful control over all the premotor circuitry that controls the mouth, some muscles of the face, and even the neck. This is the 11th or the accessory motor nucleus that controls the trapezius that gives head direction with some precision. So the amygdala, this emotional center that we usually associate with fear behavior, is very tightly connected to the control of the craniofacial systems. So to give you an idea of where we are in the brain, So if we remove the cerebellum and we go here into, that was a little fast. So yeah, anyway. So if we remove the cerebellum and look into the brain stem, this oval green area is the motor nucleus of the trigeminal nerve, where these motor neurons are located. And this kind of orange area is the premotor area, which is heavily targeted by central amygdala neurons. So the trigeminal neurons, the trigeminal motor neurons in the brainstem, they project to the muscles of the face via the ventral branch of the trigeminal nerve. So they control both the masseter, which closes the jaw and gives strength to the closing of the jaw, and the digastric muscle that opens the jaw. So these neurons control rhythmically these movements of the face, and the accessory nucleus controls head direction via its projections to the trapezius. So that the premotor neurons in the parvoreticular formation control the motor neurons and give it some sort of the opportunity for the rest of the brain to move the craniofacial system in some purposeful way. So if you imagine here that these are the motor neurons in the trigeminal nucleus, we have both excitatory and inhibitory neurons projecting to them. So in this parvocellular part that is controlled by the amygdala, we have a very strong population of inhibitory neurons tonically inhibiting the motor neurons. So that when we stimulate these inhibitory motor neurons in the brainstem, what we observe is immediate arrest of the craniofacial systems upon turning on the laser. So you can see just after the laser, the animals immediately resume eating. In the case of hunting, this is a control animal hunting an animal, a prey. But when we interrupt the motor neurons in the brainstem, what we see is that the animals, they still pursue and attempt to kill the prey, but because their craniofacial motor systems are not working, they are not able to capture and bite the prey. So they struggle with the prey without being able to kill it. You can see that the jaw doesn't open in this case, and the animals eventually give up. When we use the laser to activate these neurons, what happens is that the animals, they capture the prey, but they are not able to kill. So somehow they learned that if they wait a little bit, the laser will be off and they will be able to kill the prey. So they rest on the prey and without being able to use the jaw. But as soon as the laser goes off, they do their normal behavior. So like now, they go, they chop off the head, and they kill the prey. So this system, so we have this inhibitory system in the brain stem inhibiting the craniofacial system, and what we think happens is that these inhibitory neurons in the parvocellular reticular formation tonically inhibit the motor neurons in the trigeminal nucleus. And because the central amygdala neurons are themselves inhibitory, what they do essentially is to provide inhibitory transmission to these inhibitory interneurons here, so that the central amygdala disinhibits the motor neurons that control the craniofacial system. So it's an inhibition of inhibition mechanism. And in fact, when we deliver the light channel to the central amygdala and we place the optical fiber over these inhibitory neurons in the brainstem, what we see is basically a disinhibition of motor programs associated with feeding. So what we call fictive feeding. So when we turn on this system, what we see is that the animals will reproduce this motor program associated with feeding even if there is no food present in the cage. So they move the jaw in a way that is very similar to mastication. They have this reflex of bringing the forepaws to the mouth. But as soon as the laser is off, they again go on and wonder what they were doing, because there is no food in the cage. So I forgot. Yeah, okay. So this is a system that controls the craniofacial part of the hunting behavior, but there is a added aspect to the role of the amygdala in hunting behavior, which relates to the ability of triggering the hunting itself. So the neurons in the central amygdala project very heavily to an area called the periaqueductal gray in the brainstem. And in this area, we have some excitatory neurons that very strongly inhibit locomotion behavior. So as you can see here, we have an animal treated with saline hunting a cricket. So the cricket is trying to escape, and the animal very eagerly hunts that cricket. However, when we use a mechanism to activate these excitatory neurons in the brainstem tonically, we can see that the animals basically completely arrest their motivation to hunt the prey. And if anything, they seem very startled or scared about the presence of the prey in the cage. And this is because there is a circuit, as I said, in the brainstem, in the periaqueductal gray, where glutamatergic neurons inhibit neurons present in a second area of the brainstem known as the mesencephalic locomotor region. So this region is the region that controls the triggering of locomotor patterns in the spinal cord. So activating these glutamatergic neurons in the periaqueductal gray strongly inhibit the ability of the animals to initiate locomotor patterns. And very interestingly, the central amygdala projects very heavily to these neurons that inhibit locomotion. So again, we have a disinhibitory system where the inhibitory neurons of the central amygdala arrest this suppressive mechanism and liberate the motor patterns that control the spinal cord during locomotion. So if we do the same experiments, in this case, inhibiting the periaqueductal gray by activating the central amygdala terminals that terminate on them, what we see is that when an animal that is already very satiated sees a prey, it doesn't show much interest, but when we activate this projection, the animals very eagerly hunt and kill the prey repeatedly, even if they are not hungry. So what is very interesting is that when we trigger this mechanism, this projection from the central amygdala to the periaqueductal gray, we see animals killing prey without eating them. So they just store and accumulate dead prey in the cage. What is interesting is that this system that triggers prey pursuit and the system that triggers prey capture, these two targets of the central amygdala, the parvocellular reticular formation that the periaqueductal gray, they seem to be targeted by different populations of central amygdala neurons. So when we place tracers of different colors into these two regions of the brainstem, and we look at where they end up, we see mostly a segregation of neurons. Sometimes they terminate in different areas of the central amygdala. Sometimes they are in similar areas, but very rarely one neuron in central amygdala projects to both systems at the same time. So this led us to think that if we stimulate these two projections simultaneously, we may be able to restore the complete hunting behavior. And in fact, when we put the light channel here in the central amygdala, and we place fibers, bilateral fibers, both over the reticular formation and the periaqueductal gray, so that the animals have 4 fibers, optical fibers in the head. This, you don't really need to stimulate the whole central amygdala for obtaining hunting behavior. Just by stimulating these 2 terminals is sufficient to cause the animals to go after something that is moving in front of them and attack, grab, and bite it, and this will last for as long as the animal is being activated with the laser. We can do kind of the reverse experiment by using a combinatory viral mechanism where we have a retrograde virus injected in a target area and using a caspase directed to this virus to kill neurons in some specified way. So basically, we use this system to selectively kill neurons that project to the parvocellular reticular formation, but preserve the neurons that project to the periaqueductal gray, and vice versa. So when we use this technique, we can get rid of most neurons, for example, projecting to the parvocellular reticular formation, and we can see that it produces a very clear behavior where this is a control animal capturing a cricket, chopping off its head, and eating it. So you can see it takes some mandibular force. But then when we damage this system in particular, you can see the animals want to hunt the prey, but they cannot use the jaw properly because this system is impaired. So very interesting, they change their behavior strategy. They start using their forepaws, but still they cannot really deliver the killing bite on the prey, and the prey usually escapes, and these animals usually they are completely unable to kill their prey. So when we look at what happens when we produce these lesions in the brain, we see that damaging the craniofacial system does not impair the latency to hunt, but damaging the pursuit system dramatically increases the latency to hunt. So it's the damage to this area produces a very specific behavior impairment, where if we look at how many times they successfully try to hunt with the mouth, this only decreases if we impair the craniofacial system, not the pursuit system. And if we look at bite force itself, it's only impaired if we damage this target of the central amygdala, not the other. So we have the central amygdala producing this modulatory system where some neurons project to the periaquedutal gray and then to the mesencephalic locomotor region to control pursuit by controlling the spinal cord, whereas another group of neurons project to the premotor area of the craniofacial system and controls the neck and head muscles in a way that the craniofacial system is inhibited or stimulated. So these 2 different groups of neurons in parallel control 2 different aspects of the hunting behavior. Now, one interesting aspect of this mechanism is that in the presence of other mice, stimulating the central amygdala did not produce any obvious aggressive behavior. So when we put male animals in the presence of female, which is a kind of classical test for arbitrary aggression, what we see is that the animals, they pursue the female even before the light is on. they are very interested in the female. And when the laser is on, we see a relative increase in investigation and the oral behaviors towards the female, but never anything that looks like a social attack. So there is nothing obviously associating the central amygdala activation to attacks towards conspecifics. It seems to be relatively restricted to things that are edible, that are potential food. And the reason why that is probably is that the traditional attack area in mammals has been attributed to a specific area within the hypothalamus in the ventromedial. The ventrolateral part of the ventromedial hypothalamus was discovered about 50 years ago to contain neurons in cats and rats, and now we know in mice too, that stimulate attacks, arbitrary attacks against conspecifics. So this area contains a very small but very effective group of neurons that, when stimulated, have been shown to produce very strong attacks, including very vicious bites against opponents of the same species, against conspecifics. And to cut a long story short, what we know about this system, which obviously involves the control of the jaw as well, is that it does not seem to project directly to the brain areas known to control bite force and bite strength and the movement of the jaw. So there are no direct projections between these two areas. However, very interestingly, this attack area in the hypothalamus projects very robustly to the central amygdala, in particular to the area where the neurons that project to the neurons that control biting are located. So we have a disynaptic mechanism linking via the central amygdala the conspecific attack area in the hypothalamus to the motor systems in the brainstem that control biting and the movement of the jaw. So in fact, when we stimulate attack area neurons that terminate in the central amygdala specifically, and we measure the electrical activity of the masseter muscle that controls biting and bite force on the head, we see that this stimulation produces a very clear activation of this muscle and an increase in bite force. So somehow the central amygdala does not itself produce attacks towards conspecifics, but seems to be an important node in the system that connects attacks towards conspecifics and the activation of the relevant muscles that execute that behavior. So we have been looking at the effects of lesioning the central amygdala, specifically the central medial part of the amygdala that contains many of these neurons that control the jaw. So using some custom-made devices to measure bite force in mice, we discovered that eliminating these neurons in the central medial amygdala produces a very dramatic drop in bite force in general. So the amygdala, the neurons in the amygdala, interestingly enough, are very important for the amount of force that the animals can exert on an object that is in their mouth. So when we place these animals in the presence of a resident male, what we see is that so resident males, they attack animals very fiercely, and you can see this animal trying to escape, right? This is This is a resident, a normal resident male, attacking an invader to its territory and trying to bite it. And you can see this is a kind of a freezing behavior. This animal is basically signaling that it doesn't want to fight against the resident male, which is still is patrolling the area and trying to bite the invader. However, when we have lesions to the central amygdala, what we see is that the resident animals with lesions to the amygdala lose the ability to bite the invader and basically motivates the invader to initiate counterattacks and fight back. So that in the end, and this is a very rare phenomenon, the intruder to the cage ends up attacking the resident and leading it to submission. So a very focal lesion to the central amygdala completely changed the social dynamic between the invader and the intruder and the resident, because the lack of feedback associated with bite force led the intruder to believe that the fight could be won. And in fact, when we look at the behaviors associated with this impairment in bite force, what we see is that we see a very clear change in the behavior of the intruder. So when we lesion the central amygdala of the resident, what we see is that the number of freezing events associated with the intruder decreases dramatically. They are no longer fearful of the resident. Also, the attempt of the intruder to escape, the number of attempts to escape decreased dramatically in the presence of residents with central amygdala lesions. And the number of bites delivered by the intruder increased dramatically. So the ability of these animals to protect their territory using bite force has been greatly impaired. So we have a mechanism linking a neural circuitry associated with attacking behavior in animals to the motor system that effectuates these attacks, that basically depends on central amygdala neurons. So we have these attack neurons projecting to the central amygdala that in turn project to the premotor circuitry in the brainstem that control the trigeminal motor neurons that are those that effectuate the activity of these muscles in the head and the face. So, so here we have the parcellation of the ventromedial hypothalamus. The ventrolateral area is presumably the attack area. It projects specifically to the central medial amygdala that in turn controls these premotor neurons that are the ultimate regulators of the motor neurons of the face. So we have this hypothalamic to motor system basically mediated by central amygdala neurons. So to kind of conclude, what we think is that in many species investigated to date, homologous to the central amygdala exist in their brains using molecular markers and other types of criteria. What is interesting is the only animal so far in which these molecular markers that designate the central amygdala haven't been found are vertebrates that do not have a jaw. So we believe that the central amygdala evolved in all vertebrates that have jaws as an extension of the dorsal striatum dedicated to control these new muscles and bones that arose with evolution and modified the morphology of the vertebrate head. So it's basically, they are homologous central amygdala in basically all species of vertebrates that have jaws studied so far. Both molecularly and anatomically, they all possess a central amygdala homologue. And this includes, of course, primates, in which central amygdala neurons are very well studied, and humans, for which we know many types of responses in the central amygdala are evoked by a number of emotional stimuli, and we still don't understand exactly the connection between the conservation of these systems in higher primates and its role in behavior. Of course, early humans or early hominids were hunters, hunter-gatherers, so the hunting was part of their living, of their normal behavior, but there are a number of possibilities to interpret what the central amygdala may be doing in humans these days. So I'd like to thank some of the people that run the experiments, especially Wenfei, who has been developing all the techniques and the behavioral assays associated with this, and some other pieces of work, and some other collaborators, and those helping us with the technical aspects. Thank you.